I would like to discuss with you today how the biology of cancer cells could be used, yes, okay, can be used for non-targeted cancer therapeutics. And I will use an example, our own work on Wnt signaling, which requires, we found, membrane trafficking, macropinocytosis, multivesicular bodies, and lysosomes, all of which could in principle be targeted. And finally, I'll tell you a few things about lysosomes and signaling that in the embryo, which is quite interesting. As you know, the Wnt signaling pathway, when Wnt signals, on the top right, you see that you stabilize a protein called beta-catenin in yellow, and then that signals into the nucleus. See, well, that doesn't work very well. It's okay. It signals into the nucleus. On the left, we have a destruction complex that constantly degrades beta-catenin and contains two components such as axin and adenomatous polyposis coli, APC, that are major tumor suppressors. So when these are mutated, you get constitutive Wnt signaling. And what we have added to this is the realization that membrane trafficking is very important to maintain the Wnt signal. And so on the right, you see the formation of these multivesicular bodies, which are these little vesicles that take the receptor from the membrane together with the destruction complex and these, in red, the enzyme called glycogen synthase kinase 3, which is what degrades normally the beta-catenin. So the beta-catenin is stabilized. And on the inset, you see a multivesicular bodies. When Wnt is activated, the GSK3 translocates into these cells. So you would say, well, this is very complicated that you need this for Wnt signaling, but in reality, this is just taking together the normal membrane trafficking machinery that all cells have. So here you have on the big vesicle there, you have the outside-inside vesicles forming multivesicular bodies that go into the lysosome. There's going to be another one being formed here. So that's where the GSK3 is going to go. And in this case, it's multivesicular body is going to go to the outside and form these so-called exosomes. So if we just take a HeLa cell and we treat it with Wnt protein on the outside, within 5 to 20 minutes, you see on the right that there are these big vesicles being formed. These are multivesicular bodies that will go on to form lysosomes. And we know this because we can add bovine serum albumin with a fluorescent marker, which when it gets to the lysosomes fluoresces as BSA dequenched. And when we see on the right side, on the left side, a cell without Wnt, a group of cells, and then we add Wnt to that group of cells, and you see that within 30 minutes, you have taken up much more. You have, if you look at the lower right, you see, I hope, more red. That means that the cells like to eat the BSA and digest it from the outside. And the lysosomes become more acidic with this the membrane trafficking. And so this is a lysosensor. In panel A, you see the pH is about 6. But when you add a GSK3 inhibitor in panel D, you have much more acid lysosomes. And the way, so therefore, Wnt GSK3 regulates the activity of the lysosomes, and a cell on Wnt is feeding on extracellular proteins preferentially compared to a normal cell. So these are other points that you can target. And the way these cells get in, this through macropinocytosis. Pinein means to drink, and the cells drink this, in this case, dextran. So on the left, you have a without Wnt. In the middle, with Wnt, you're taking up the dextran. And then on the right, if we put an inhibitor called IPA, it's an inhibitor of a sodium proton exchange protein. that blocks macropinocytosis, which is an actin-driven process, and it's repressed by GSK3. So if you put IPA, you inhibit Wnt signaling as well. So for the cell, this is a very great effort to when you get Wnt, and you will see here on the right panel, we are going to now add Wnt to a cell that is stained for actin, and you will see that very rapidly this tries to form these vesicles on the outside. And this happens not only if you add Wnt, but also when you have a mutation in this tumor suppressor called axin-1. This is a hepatocellular carcinoma cell line. And on the left, we've added back axin-1, and you see there are no more macropinocytosis being formed, although the cell has been in culture for 40 years. It's called the Alexander cell. And so you see now the effort that the cell is putting in eating from and taking all this liquid from the outside. And so that's a vulnerability for the cancer cell. So this is an axin-1 mutation, but you can also, of course, the APC is the main tumor suppressor in colon cancer. And when we have colon cancer in panel A, it has nuclear beta-catenin colon cancer, but we can then add in panel C, we can add IPA, which is this inhibitor of macropinocytosis, or even better, amiloride, which is a commonly used diuretic, amiloride, which blocks the same pump. So that is used for decades in human use also. But so then therefore, even constitutive Wnt signaling can be inhibited if you inhibit macropinocytosis. And all this took place in only 2 hours of treatment. So just 2 hours and down comes the beta-catenin. And we can also do the converse experiment, which is to increase the macropinocytosis. And the macropinocytosis can be increased, as you know, with what are called tumor promoters. The tumor promoters don't cause cancer in themselves, but they promote the development of cancer. It's known since 1941, and that multiple applications of this phenocopy the effects of an initial carcinogen. You put a carcinogen and then you paint, for example, a rabbit ear with croton oil or turpentine, and the active agent is called phorbol ester, which mimics deacylglycerol, and it was known from the work of Joel Swanson that this increases macropinocytosis. So we can increase macropinocytosis with a tumor promoter. And so when you see on the right, when you add to these cells that on the left, they have nuclear beta-catenin, so they're constitutively Wnt, but they will get even much more beta-catenin if we just add the PMA. This is a 24-hour treatment, but within 1 hour, you can see this going up. So this causes an increase in macropinocytosis and an even further increase than the absence of the APC. And we do know that in human cancer, the more beta-catenin, the more malignant. So if we look in this slide in the middle column at the bottom, you have, these are histological arrays. The adenocarcinoma in C prime has a lot of beta-catenin. And interestingly, on the left panel, if you look in green, you have GSK3. In normal colon, there is much more GSK3 than down at panel C, which the more malignant, the less GSK3, in agreement with our model that the GSK3 goes into the lysosome where it is degraded. And furthermore, if we now look at lysosomes themselves, the more malignant the tumors, if you compare panel A to the panel C on the bottom in red, that's stained for the V-vacuolar VATPase, which is the enzyme that acidifies the lysosome. So therefore, one can, if you inhibit lysosome activity, you can provide, if you could provide a target to prevent cancer progression. And indeed, other people have used in xenografts, if you put in an inhibitor of VATPase called bafilomycin, you can decrease the growth of the tumor. Now, so therefore, no, there are many places where one could target these things, and I'm interested in this because of the frog embryo, which we work with, and if you look there, there are 4, 8 cells, and as they develop, if you see there is an asymmetry in which one side of the embryo is less pigmented than the other. The less pigmented side will become the dorsal, the back. The more pigmented will become the belly, the ventral. And if we cut a section through this embryo and you see that on the dorsal side, you find these very interesting multivesicular bodies. There you have one with 3 intraluminal vesicles, one vesicle being formed with a clathrin coat, it's called, and then all these other vesicles around it that are presumably bringing in the GSK3 to be sequestered. We don't know that the GSK3 is in these multivesicular bodies, but we do know that on the dorsal side of the embryo, you have, if we look, for example, at P prime, there we have more lysosomes marked with a marker of active lysosomes called SIR lysosome that stains the active beta-catenin. So this is at the 64 cell stage, you see the less pigmented side has more active lysosomes. And we have defined conditions in which you can take this inhibitor of the VATPase of the lysosomes. At the 32 cell stage, you put 7 minutes, and you see you knock out the development of the head in the endogenous frog embryo, and it off goes most of the brain. So you need these lysosomes for the endogenous development of the embryo. And so head formation requires lysosomal acidification in the embryo. And furthermore, we can do the converse experiment. We can increase macropinocytosis with forbolester. You remember? So if you look at the 2 left panels, the forbolester doesn't do much or nothing to the embryo if you inject a little bit. And in the top right, we inject a little bit of lithium chloride, which it mimics the Wnt signal. It gets a little bit of a bigger head. But if you put lithium and forbolester, you see you get an embryo that has a radial head. No trunk, it's all heads, meaning it potentiates enormously the forbolester, the effect of Wnt in the embryo. So what happens from this and work of many others is when you have the sperm entry, there is a cortical rotation, and we propose that there are lysosomes and multivesicular bodies being formed on the dorsal side, and that's the initial Wnt signal that produces the axial development of the embryo. Now, so this is about what is interesting about the development of the embryo, but the implications for cancer are that potentially interfering with macropinocytosis or multivesicular bodies or lysosome activity or other aspects of membrane trafficking. For example, you can treat with inhibitors of the VATPase, of which there are many, of the sodium proton exchangers, or extracellular macromolecules that are taken up by macropinocytosis. And this could provide non-targeted, sort of non-specific, wide-spectrum inhibitors of cancer proliferation or of cancer progression, like the tumor promoters. However, the truth is that only the targeted therapeutics have been successful so far. These would have to be used in combination, so I'm very happy that our next speaker will tell you about the successes in targeted, so this is non-targeted. And with that, next speaker being Charles Sawyers, my friend. And so then with that, I will thank my colleagues, and I will thank you for your attention. Thank you.